Assalamualaikum students. Before going to our educational topic, please subscribe my channel Muntazir Mehdi Science TV on YouTube. Press the like button and share this lecture to your friends and classmates who have no access to it to add in their science education. And those students that are preparing privately can benefit from our channel very effectively. Let's come back to our topic. Today our topic is biotechnology. First we see its definition, then we would explain it with the help of other explanations. The definition of biotechnology is that the account usability of living organisms, their systems or processes in the manufacturing of different types of factors, proteins, genes, DNA fragments, DNA fingerprinting, and many other applications in different fields of life and in services industries is called biotechnology. Biotechnology has different types of advantages as well as disadvantages. First, we see the advantages of biotechnology. When Mendel's work redesigned in 1900, then after its rediscovery, the geneticists have originated many advanced techniques. In this way, a new era of DNA technology started. Biotechnology has many different types of uses. Some are described as: first advantage of biotechnology is that genes can be taken out from one organism with the help of modern techniques. Then these genes are implanted into another organism with the help of similar modern techniques. So these genes produced a wanted substance such as insulin. During prior era, the insulin dependent diabetes mellitus patient received the insulin from the dead animals, but today they receive human insulin with the help of recombinant DNA technology, that is the product of biotechnology as well as genetic engineering. You can watch the recombinant DNA technology at our channel in detail. Biotechnology has also produced different types of drugs and vaccines to control human afflictions. Instead of synthesizing insulin or other factors, genetically engineered bacteria are also used to clean up environmental pollutants. These genetically engineered bacteria are used to clean up beaches where oil pollutants are. These bacteria increase the fertility of the soil and also kill insect pests. Biotechnology is now used to change the genotype of different organisms. Not only genotype, but it can also alter the phenotype of different plants or animals. In addition to plants and animals, gene therapy is also under clinical trials in humans. Gene therapy is used to repair a malfunction gene in those persons that carry such a malfunction gene. Biotechnology not only possesses advantages, but it also has some disadvantages, such as some people oppose the manipulation of genes, collaboration of genes for some senses, for some generalization. So far, harm effects of biotechnology have not been discovered, but the agitation that there will be health and ecological consequences of biotechnology in the future. Next, we see the definition of gene cloning: the creation of a large number of same copies or uniform copies of a gene is called gene cloning. In other words, we can also say that the production of huge amount of identical copies, similar copies of a gene is known as gene cloning. A very large quantity of a gene can be produced by recombinant DNA technology. But another method for producing different types of genes copies is also present, that is known as polymerase chain reaction, which produces a lesser number of gene copies as compared to recombinant DNA technology. Polymerase chain reaction carries out in a battery test tube. Shortly, we can call it as PCR, as I have already prepared two lectures for recombinant DNA technology and one for cloning. So today my focus will be at polymerase chain reaction. So now we will understand the polymerase chain reaction, that is shortly called as PCR. Now we see what is polymerase chain reaction, why we do PCR carry out. Polymerase chain reaction is a particular technique, a specific technique during which large number of a DNA segment or a large number of identical copies of any DNA segment or a large number of identical copies of a gene can be synthesized. For the formation of huge number of these identical copies, a particular enzyme takes part in this process PCR, that is known as DNA polymerase. In other words, we can say that a particular DNA piece or a gene can be amplified with the help of PCR. PCR is an artificial process during which we amplify only a segment of a DNA or a gene in a test tube or in a pind of tube, whatever an artificial tube is result in an artificial chamber or in a artificial tube. So these fragments of the DNA or fragments of the genes are present only in their single state that have no either a start codon or stop codon. There is no need for a vector because it is an artificial process. Polymerase chain reaction was first introduced by a biotechnologist, Cary B Mullis, for synthesizing multiple copies of a DNA fragment or a gene artificially. This technique gives a large number of DNA fragments of genes or a single gene quickly in a test tube, because earlier methods of getting multiple copies of a specific sequence of DNA fragment were time consuming, delaying, as well as expensive. But it is an easy and a cheap technique for obtaining a large number of copies of specific sequence of a DNA fragment or a gene. PCR is very specific, easy and a cheap way to use because it can create millions of copies of a single gene or any special piece of DNA molecule sharply in a test tube. It is a very unique technique during which the targeted DNA sequence can be less than one part or one smaller part a million of the total DNA sequence, which means that any single human gene such as insulin gene, a smaller piece of a DNA molecule, can be amplified again and again by using polymerase chain reaction. PCR requires following materials. First one is DNA polymerase. The name of the polymerase chain reaction comes from the common enzyme DNA polymerase. DNA polymerase is that enzyme that is used during the DNA replication process in any cell. You must keep this thing in your mind that PCR is a chain reaction. So the DNA polymerase is involved in the carrying out DNA replication again and again. So millions of copies or millions of replica of the required DNA fragment or required genes are produced in a short period of time. Second required material is primer DNA. PCR essentially needs a primer DNA for starting its polymerase chain reaction. This primer DNA consists of sequences of about 20 nitrogenous bases. As we know that these are nitrogenous bases that are present in a double-stranded DNA molecule. So when 20 nitrogenous bases combine to each other, then it forms a primer DNA. This is that primer DNA which is needed for initiation of polymerase chain reaction. These 20 bases are complementary to the nitrogenous bases on either side of the target DNA molecule. Complementary means these nitrogenous bases are able to attach on either side of the DNA, as their attachment is according to the universal rule which says that adenine always forms pair with thymine by means of 2 hydrogen bonds, whereas guanine always forms pair with cytosine by means of 3 hydrogen bonds, and vice versa. Maybe you have already know that it is a universal rule that the attachment of adenine will be possible with the thymine, whereas the attachment of cytosine will be possible with the guanine, and vice versa. If these nitrogenous bases would be attached with the opposite nitrogenous bases instead of their complementary base pair, then it will lead to mutation. So replication process is not initiated by the DNA polymerase enzyme. Therefore, primers are required for initiating the DNA replication. So when once DNA replication is started with the help of primer DNA, and DNA polymerase comes and takes part to continue the process. Then primer DNA attaches with the complementary base pairing of the target DNA strand, and DNA polymerase begins to copy the target DNA. Third participating material in the PCR is high temperature. Enzyme DNA polymerase is thermostat, which means that this enzyme can withstand at very high temperature and can retain itself. So it is a temperature insensitive enzyme because it is obtained from the bacterium Thermus aquaticus. These bacteria live in the hot springs. That's why DNA polymerase can bear very high temperature nature of these hot springs. DNA polymerase is also known as Taq polymerase. Now we understand the PCR mechanism with the help of a theoretical diagram. As we know that a DNA molecule is present in this double helical spring-like form, but when high temperature is applied to this DNA molecule, such as heat for 1 minute, then it causes to separate its both strands like this. When it is provided with heat, then both of its strands get more separated apart. One strand is from 5' to 3' whereas the second strand is from 3' to 5' direction. It is just only a segment of DNA, not representing the whole DNA. As we can see that DNA molecule is denatured, whereas DNA polymerase can withstand. Therefore, DNA replication is not interrupted for polymerase chain reaction, and additional enzymes are also not required for this medium. It is a test tube. So after the separation of the both strands of a DNA fragment, now these are cooled for about 2 minutes. Then primer DNAs are added. After that, DNA polymerase enzyme is added and wait for about 1.5 minutes. Finally, we get millions of copies of a small DNA piece or a single gene. This complete cycle is repeated again and again and again. Fourth material required for polymerase chain reaction is a PCR machine. In these days, polymerase chain reaction is done with the help of an automatic machine that is known as PCR machine. It is also called Thermocycler. As polymerase chain reaction is repeatedly done, so it is a cyclic process, chain process, and it requires high temperature. So PCR machine is also known as Thermocycler, that is short by its name. It is an equipment present in the at the end. We will see a major difference between the polymerase chain reaction and gene cloning, because polymerase chain reaction cannot replace the gene cloning. Gene cloning is used for the production of a large amount of protein or genes. On the other hand, PCR is used for the synthesis of genes in smaller amounts as compared to gene cloning. So students, it was our topic today, biotechnology and polymerase chain reaction. I hope these videos will have been benefited to you. So make sure to subscribe our channel for receiving notifications of further upcoming videos, and also make sure to like my videos. I will advise you keep your best attention to your education. It is the only way for you that you can service your country. Thank you for watching our channel.